All right, let's talk about how carbon dioxide is carried in the blood now. So, we've talked about oxygen. We talked about how oxygen has a relatively low solubility in water. Carbon dioxide is a little more soluble, so you can fit a little more carbon dioxide into the plasma as it is anyway, for the same PO2, for the same partial pressure. But carbon dioxide does something in addition that actually really changes a lot about how it interacts with the blood. And that's that it reacts with water in what we call the bicarbonate buffer equation. This has a lot to do with how the body regulates pH. We discussed this somewhat in lab 20, but in case you haven't seen that yet, let me go over how this works. So, carbon dioxide, CO2, can react with water, H2O. That reaction happens reversibly. That's what this double-ended arrow means. It can go either way, to form something called carbonic acid, H2CO3. That, in turn, can split up into H+, which we usually call proton, and HCO3-, which is the bicarbonate ion. So this is carbonic acid, proton, and the bicarbonate ion. Now, that means that if I put carbon dioxide in water, some of that CO2 will react with water to form carbonic acid, and that carbonic acid will tend to split up and form proton and bicarbonate. This is the full system, however, and we're going to talk about it a little bit more, and then we'll talk about how we usually simplify it. Now, how much of this proton you have in solution determines the pH, acidity versus alkalinity of a solution. The more H+ you have, the lower the pH, which is more acidic. So that means if I put CO2 in water, that ends up producing protons, adding them to the solution, which means that adding CO2 to water makes the solution more acidic. Technically, CO2 is not an acid, but it reacts with water to form an acid, carbonic acid, which then acidifies the water. So putting CO2 in makes the solution more acidic. Now, each of these steps can technically reach an equilibrium. So if I put some CO2 in water, it'll form carbonic acid, but carbonic acid can spontaneously turn back into CO2 in water. So if I put CO2 in water, it will do this until there's enough of this reacting backward to have an equilibrium, where it's going this way just as much as it's going that way. Likewise, this one also has an equilibrium. Carbonic acid splits up into proton and bicarbonate, but they can recombine to form carbonic acid. So really we end up with sort of a three-way equilibrium here. Now, this one isn't very interesting to us, and there actually isn't very much of that in the solution. It doesn't stay in this form very long. So usually, this gets simplified to just be CO2 and water react to form proton and bicarbonate. We skip that middle step. And we talk about this being an equilibrium, where if I put CO2 in water, I get proton and bicarbonate, but some of that comes back to form CO2 and water, so I end up with a balance between these two. Now, that first step, CO2 and water to carbonic acid, that is actually a slow reaction on its own. It happens spontaneously, but relatively slowly. So, in the body, it has to happen a little bit faster in order for us to see this happen effectively. Now, there's an enzyme called carbonic anhydrase, which catalyzes that reaction in either direction. And it's a very, very effective enzyme. It means that CO2 and water can very easily turn into carbonic acid, which becomes proton and bicarbonate, and vice versa. So in the presence of carbonic anhydrase, these things go back and forth pretty much freely, without any real delay. In the lab 20 background, I believe that's where I tell a fun story about how I personally experienced this. It involves deep dish pizza and dry ice. But that's a story for a different time. Now, for this, that means that in the presence of carbonic anhydrase, these are pretty much freely interchangeable. So when I put CO2 into my blood, some of it will react with the water to form proton and bicarbonate, which acts to acidify the blood and add bicarbonate ions to it. So in other words, more CO2 turns your blood more acidic. Now this carbonic anhydrase is not just floating around free in the plasma so much. It's actually mostly located inside red blood cells. So that means, by the way, keep this in mind, because this plays a huge role in how our body... Well, we need to really talk about that right now, don't we? Now, this plays a big role in how we regulate ventilation, which is part of how we regulate the pH of our body. Since CO2 will acidify the blood, it can go the other way too. If I remove CO2 from the blood, some of the proton in my blood, which is making it more acidic, will react with the bicarbonate to bring the equation back this way, removing H+ from solution, making the blood more alkaline. So by regulating how much CO2 I have in the blood, I can regulate the pH. If I hyperventilate, breathe much faster than I need to, so like that, I'm getting rid of CO2 faster than my body is producing it. By removing CO2 overall from the blood, I pull the equation this way, removing protons from the blood and making it more alkaline. And actually, the lightheadedness you might feel after hyperventilating comes from that. It comes from the pH change in your blood. Likewise, if I hold my breath, more CO2 builds up. That extra CO2 reacts with water to add more protons to solution, and my blood becomes more acidic. Now, there are some things that buffer that a little bit. So we'll talk about that in a little bit. But that is one way that my body regulates the pH of my blood, by regulating my ventilation. We're going to get to that in the last part of this lecture. But back to this. The carbonic anhydrase that we're talking about here is not just free in the blood. It's mostly found inside the red blood cells. So where I want to go next with this is talking about how CO2 interacts with the red blood cells and how it is actually carried in the blood. Because it actually shows up in several different forms, and we need to talk about how it moves between those. So I'm going to erase this and switch to putting a drawing of a red blood cell on here. In that red blood cell, I'm going to have, this will represent hemoglobin, Hb. Now, also on this red blood cell, I'm going to need to put a particular antiport on here. This is a bicarbonate chloride antiport. It's a passive transporter that allows bicarbonate to cross the membrane in one direction as long as chloride ions are crossing the membrane in the other direction. That's going to be important in a bit here. And finally, I need to add in here that we have the enzyme carbonic anhydrase. So now let's follow the journey of some carbon dioxide that we add to the blood. In the blood I have CO2. Now this CO2 can go through cell membranes like all these dissolved gases. So CO2 can easily pass into the red blood cell. Once it's in the red blood cell, there are actually two things that can happen to it. One of them is that CO2 can bind to hemoglobin. This is something we haven't talked about yet. Hemoglobin, this protein which we've talked about binding primarily oxygen, can also bind carbon dioxide. We mentioned this a little bit in the previous lecture where we talked about the oxygen-hemoglobin dissociation curve and things that can cause it to shift. Carbon dioxide binds to hemoglobin not on the iron. It doesn't compete with oxygen. It actually binds to the amino terminus of each of the 4 subunits. It binds to the protein part, forming something called carbaminohemoglobin. Carbaminohemoglobin. Great word. So hemoglobin can hold on to that carbon dioxide. It's not permanent. It can come right back off. But right now we're going to say some of the CO2 that comes in here sticks to the hemoglobin. That affects how it binds oxygen. It tends to cause it to release a little oxygen, but we'll leave that alone for now. The other thing that can happen to carbon dioxide is that it can react with water to form two things. proton and bicarbonate. That was the part of that bicarbonate buffer reaction. Now the protons, as it turns out, tend to stick to the hemoglobin, which is good for us. As those protons get stuck on the hemoglobin, they don't end up acidifying the blood plasma. So this minimizes how much CO2 changes the pH of the blood. By buffering these protons, we limit how much acid we add to the blood. The bicarbonate can leave this cell in exchange for chloride coming in. So that means that we have three places that carbon dioxide can end up. Some of it can stay out here in the plasma. Some of it ends up bound to the hemoglobin. And some of it ends up in the hemoglobin in the form of bicarbonate ions rather than carbon dioxide. Now, since each of these is reversible, CO2 can come in and bind to the hemoglobin or come back off. It can react with water to form bicarbonate or bind back up to the proton and turn back into CO2 or go back out of the cell. You can move back and forth between these. This is another sort of equilibrium. We could almost imagine it as CO2 in the plasma can move back and forth between CO2 on hemoglobin, which can go to, actually this is sort of more of this way. CO2, actually, sorry, not CO2, bicarbonate. It's a three-way equilibrium like that. And so we tend to reach a particular balance between these three states, based on how easy it is to go from one state to the other. In the end, that balance we tend to strike is that if I put 100 CO2s into the blood, I end up with 7 of them. 7% of it is found in this form at any moment. 23% is found on the hemoglobin at any moment. And 70% is found as bicarbonate ions in the blood. Now, any individual CO2, you can't say which one it's in. So at some moment, I've got a CO2 out here like this. The next moment, it might come in and stick to the hemoglobin. And then one on the hemoglobin might come off and move out here. And then one in there might come through and turn into bicarbonate. They can flow back and forth, but this is roughly the balance you find. You could imagine it as if you took 100 people and said, okay, 7 of you sit down, 23 of you lie down, 70% of you stand up. Now you can switch back and forth, but if one of you goes from standing up to lying down, somebody who's lying down has to switch state. And that way you maintain a balance. In any case, this balance follows the rules of equilibrium, what we call that law of mass action, where if you add a bunch of stuff to one state, it tends to push things into the other states until you get back to that balance. So that means that if, for example, I add CO2 to the blood, some of that CO2 tends to come in here and stick to the hemoglobin and turn into bicarbonate until I get back to the 7-23-70 balance. Likewise, if I take CO2 out of the blood, some of the bicarbonate tends to come back in and turn back into the CO2, and some of the stuff on the hemoglobin comes off and turns back into CO2 to restore that balance again. I've got a way of demonstrating that I'll try to show you, I believe, at this point. Let's begin by imagining that we have some blood about to enter a systemic capillary. On this graph, I've imagined that we have 10,000 carbon dioxide molecules in that bit of blood, and I've distributed them among the appropriate areas. So we have 7% in the plasma as CO2, that's shown in orange, so that's 700 molecules. 23% bound to hemoglobin, that's shown in yellow, so that's 2,300 molecules. And 70% are in the plasma as bicarbonate ions. That's 7,000 molecules, and that's shown here in green. Alright, so let's take that blood and imagine that we now enter the systemic capillary. Outside the capillary, we have tissue and its extracellular fluid, And that has a relatively high concentration of carbon dioxide. We'll show that here in blue. Now remember that carbon dioxide levels in the ECF won't change, since that tissue is continuing to make CO2 as a waste product from aerobic metabolism. So that size bar isn't going to change at all during this. So now let's imagine the CO2 entering the plasma, bringing the plasma CO2 up to match the CO2 of the ECF. So we just brought in 1,300 molecules of CO2 from the tissue in that process. So now they're both at 2,000. If that CO2 stayed in the plasma, that would be all we could take. See how the plasma is now at equilibrium with the ECF, since their bars are the same height. But now our 7%, 23%, 70% balance is off, because now more than 7% of the CO2 in the blood is in the form of CO2 in the plasma, rather than hemoglobin or bicarbonate. So some of that CO2 in the plasma now has to move to the hemoglobin and turn into bicarbonate. So we take the total CO2 in the plasma, hemoglobin and bicarbonate all together, and redistribute it into a 7:23:70 ratio. But that gives us room for more CO2 to move in from the ECF. So we move more in, and now we redistribute it into the 7:23:70 ratio. From here on out, we can skip the whole step of bringing the plasma up to equilibrium, and we'll just watch as the CO2 we bring in distributes itself among plasma, hemoglobin, and bicarbonate. And that process continues until eventually we get to the point where the plasma CO2 matches the ECF and the CO2 is in the right ratio in the blood. At that point, we can't take any more CO2 from the tissue. So we're done. Now in that process, since we had places to put the CO2 coming in, we absorbed a lot more than just 1,300 molecules. The total amount in this simulation, if we started out with 10,000 in the blood total and then brought the tissue up to 2,000, instead of 1,300 absorbing, we brought in a little bit under 18,500. Now that same process would work in reverse when we got to the pulmonary capillaries. CO2 would go from the plasma and go into the air, so that would make the plasma portion go down. Then CO2 would come out of the bicarbonate and hemoglobin areas and go into the plasma. That would move into the air, and that would repeat itself. You'd get something similar when oxygen enters the blood in the pulmonary capillaries. It's related, but it has some differences. In that case, oxygen enters the plasma, and then that gets picked up by the hemoglobin, which takes it out of the plasma, which allows more to move in from the air to the plasma, and that goes to the hemoglobin, and so on. All right, so now let's talk about what you might observe. So imagine I'm in the tissues, and CO2 is entering the plasma from the cells. As that CO2 enters the plasma, it's going to move in here, some more is going to bind to the hemoglobin, and some is going to come out through here, and I will end up adding more bicarbonate to the blood. That will force us to bring chloride in. So in the tissues, as CO2 is entering the plasma, you could follow this through and say chloride will be entering the red blood cells in the systemic capillaries, where we're adding CO2 to the blood. Likewise in the lungs, when CO2 is leaving the plasma going out to the air, think about what's going to happen to the chloride. As CO2 leaves this state, some of this will come back in to replenish it, which means chloride will have to be going out at that point. So in the pulmonary capillaries, we expect to see chloride leaving the cell rather than entering. That's a phenomenon called chloride shift. It's kind of an interesting thing.